Hi, this is Miss Litton, and this is the day before a Unit 2 exam. Couple things I want to go over with you. First of all the essays have been discussed and posted on YouTube. So I'm not going back through the essays again today because I had dates set aside for that. It's already been discussed. Go back. If you couldn't come on those days, go back and look at that. We've also done individual chapter reviews as well, and I encourage you to go back and look at that. This is a day of kind of big picture, stitching it all together, and if you have any last minute questions, because technically when would we have had this test? Last week, yeah, last Friday. Okay, so couple things. Chapter 5, membrane structure and function. So I'm encouraging you to be confident in that, know the essay on it. But when I think about membrane structure and function, the kinds of things I would make sure I knew, okay, is how a membrane is built, the 4 components of a membrane, what role each of those components play in the integrity of that membrane and the functioning of that membrane. Did you hear all that? And I would have the big idea of how things can get across a membrane. It can go through the what? Phospholipid bilayer, but not if you are large or charged. Good. Or it could use a what? Channel or a carrier. But then that makes me think, when you learn the chapter on membranes, we don't just talk about transport, but we talk about other roles that proteins play. I would be comfortable with that. I would know all the roles. Let's review all the roles that proteins can play. They can be what? Channels, carriers, cell recognition, receptors, enzymatic, and what? Cell-to-cell junction. I would be able to explain that, okay, and maybe give an example of each one of those. Okay, so we said through the phospholipid bilayer. Then what's the second way they can use? Channels or carriers, and then what's the third way? Endo and exocytosis. Perfect. In my mind, big ticket items is whether a form of transport is passive or active, okay? And I would know who would fall underneath those categories. When you're talking about passive transport and through the phospholipid bilayer or using an aquaporin, what are we talking about? Water, right? Oh, yeah, small, non-charged, yeah. But diffusion is just something going from a higher concentration to a lower concentration. But osmosis is the diffusion, water across a semi-permeable membrane. 3. Yeah, diffusion, water, semi-permeable membrane. When you start talking about that, then you need to start using words like hypertonic, hypotonic, isotonic, right? And water must flow from the hypo. You're using words like osmotically active substances, right? Something that has, if you have rocks or sand in a jar, that's not going to do anything, right? It has to be something that's going to interact with that water, like a salt or a sugar, right? Bound water of some sort. And you would talk about water potential, okay? Because you're going to go from a higher water potential to a lower. We start talking about osmotic pressure to have something come in, right? Then you have to start talking about, if we're talking about cell membranes, we can talk about animal cells, but we level up when we start talking about what? Plant cells, because plant cells have a cell wall that can push back, right? And then you start talking about things like water potential. And remember, water potential is a factor of 2 things. What 2 things? Solute potential and pressure potential. And we did a lab, right? And in our lab, we, and you have that lab back, we talked about, do I have this frozen? No. Remember, in the lab, the first thing we did is we modeled weirdness. Un momento. I'll just use my finger. Okay. We did a model. Okay, and that model we modeled using, what was the first thing we did in our diffusion lab? Yeah, and we had a beaker, remember? Oh, this is going to drive me nuts. And in that beaker, remember we put iodine and water, and then we put a baggie, and the baggie had what in it? Starch and glucose. and we modeled something being semi-permeable, because some things could pass through the dialysis tubing and some things couldn't. So that was to model the idea of things being semi-permeable. We knew that the bag was permeable to iodine because we had evidence. When iodine moved, it was outside the beaker. The only place it could move, outside the dialysis tubing, the only place it could move was in. And if it moved in, then how would we know? The bag would turn dark. And we knew starch didn't leave. Starch was too big to leave because, and then she starts up again. Starch was too big to leave because if it went out, then the whole beaker would have turned dark blue. Yeah. So that's what that first part was. Then the second part is we were looking at water potential, right? So first part was size matters, right, for how it could move through the semi-permeable membrane. And then the second thing we talked about was water potential, because you had different molarities of solutions, right? And then you were able to measure, or predict that the higher molarity, that bag would gain the most weight. And there's an essay question about that, right? If I have unknown solutions, and I know there are these molarities, but all my labels fell off my jar, could I design a lab to tell me which solution is which? Could you answer that question? Good, because that would be important to answer. So then we talked about instead of modeling and just using like a dialysis tubing, we used real cells. What were the real cells we used? Potatoes. And we were able to determine the potato's water potential by finding out the mass where it wouldn't gain or lose weight. And because where it wouldn't gain or lose weight, which we didn't know for sure what solution, we inferred that from our data, right? By putting in increasingly concentrated solutions, we inferred from our data, by drawing a line from our data, this is where it wouldn't gain or lose weight. So it's at that point that our pressure potential was zero. And as soon as I had my pressure potential taken out, that was one of my unknowns taken out of my equation. So now I knew water potential just equaled solute potential. And I could calculate for solute potential. And can you calculate for solute potential? If I gave you that, would you know how to do it? Make sure you know how to do it. You see my eyes? Okay, make sure you know how to do it. And then the next part was talking about active transport, right? Using a protein because it was with energy. But if it was passive transport, could you use a protein? Yes. What would that be called? Facilitated diffusion, right? And we talked, what was a good example of active transport using a protein? Sodium potassium pump. Good. 3 sodiums out for 2 potassiums in. And then the last part was endo and exocytosis. And remember we had 3 different types of endocytosis. Do you remember what those are? Pino, phago, receptor mediated. And then exocytosis is just, okay, stop throwing that around, you're gonna drive me nuts. Exocytosis is when it left the cell. Okay? So I went a little bit more on that particular chapter just because it was the farthest away and you might have forgotten some things. Again, all the essays, those are already recorded and posted on YouTube. Any big ticket items on chapter 5 that you just like, I tried to hit all of the big tickets. We good? Okay. The next part, metabolism, energy, and enzymes. There's a lot in there that sets up both chapter 7 and chapter 8. The first part we talked about, and remind me if I go off track because I'm trying to just remember everything we talked about in class. We talked about the types of energy, right? Like kinetic energy, right? And then we talked about, I can't remember my flow of this, types of energy, and then did we start talking about ATP? Am I jumping around? Should I look? Let me look. Oh, entropy, yeah. Okay.
And then like free energy.
Yo, yeah. Okay. Let me get to this. Okay. Don't forget your highly suggested reading and thinking. Do you hear me?
Yes.
Don't forget your highly suggested reading and thinking. And any question that has a list on it, where I could say list off this, know those. Okay. So, yes, we talked about the laws of thermodynamics. Make sure you know the difference between them. What does the first law say? Energy cannot be created or destroyed. It could just change forms. But the second law says every time it changes, transfers, you lose some as heat. Good. Then we talked about entropy, and that's the amount of what? Disorganization. Disorganization. As disorganization goes up, potential energy goes down. Good. Potential energy goes down. So exergonic reactions, does entropy go up or down? Down. Exergonic. Down. You're releasing energy. Releasing energy was contained potential. Now we've released it. Entropy goes up. Make it make sense to you, not memorization, right? If it's exergonic, I'm taking some amount of energy and I'm releasing that energy out. Contained energy, dispersed energy. Not as useful when it's dispersed, right? More potential energy, less potential energy. Less entropy, more entropy. It's a mess all over the place. Does that make sense? Make it make sense to you. Do you need me to go over it again? Is there any questions in your mind? Yes? Do we need to know the equations for Gibbs reactions? Any equation that you need to know is provided for you. Yeah. Okay? Okay, then we started talking about reactants and products of metabolic reactions. We talked about free energy. I just went over that. We went over ATP, the structure of ATP, adenosine triphosphate, the third phosphate. We've learned all about that. We talked about metabolic pathways. We talked about catalysts in general make an enzyme go, or make a reaction, not an enzyme, but a reaction go faster. And if it's an organic catalyst, we call it an protein-based. If it's coenzyme, if it's organic and not protein, right? Yeah, we call it an enzyme, right? If we have a protein-based catalyst, it's an enzyme. But if you have some organic attachment you need to put onto it, it's called a coenzyme. Okay, coenzyme. Then some significant things about enzymes. Their shape, right? We did a whole lab on this one, right? Our enzyme lab. The shape of that enzyme is critical to its functionality. Why does it have that shape? Because enzymes are made out of proteins, and we know it's primary, what? Secondary, tertiary, quaternary structure. And that completed structure, that final structure, that conformational shape in space, that's going to have a what site that's critical to it? Active site. If you do something that alters the active site, you alter the functionality of that enzyme. And we looked at things that can alter enzyme activity. And one is anything that changes the active site, binds to the active site instead of the substrate, binds to another part of the enzyme, which ends up changing the allosteric site, changing the active site. Those can all affect. Inhibitors, right? We had competitive and non-competitive. What would be the one that actually binds to the active site? Competitive. Binds to a secondary site. Non-competitive. Non-competitive. And we talked about, in our lab that we did a lab on enzyme. We said the amount of what you have can affect the amount of product. The amount of substrate. And if we increase the substrate, then we were filling more active sites, which meant we could do more of those reactions, which meant we got more product. I would know how enzymes can be regulated. That's an important thing because enzymes mediate every metabolic reaction, right? So that's important for life. So things that regulate enzymes would be really important. Did you see my eyes? Really important. I would know. Okay, then we went into photosynthesis and cellular respiration in general. And to do that, we needed to talk about things being oxidized and things being reduced. Okay, so who has more energy? Reduced. How do I get reduced? By the addition of hydrogen ions. Addition of hydrogen. Give me some more. Loss of oxygen or the addition of electrons. That's how I get reduced. If I am reduced, I'm at a higher energy level than if I am oxidized. How do I get oxidized? Lose hydrogen, gain oxygen, or lose electrons. Good. We call them redox reactions because every time one thing gets reduced, something else must be getting oxidized. When that happens at the same time, what kind of reaction can we call that? It's a coupled reaction. We can talk about endergonic and exergonic reactions being coupled reactions. The energy released from one goes to the other, right? And in an oxidation, the energy released from one goes to another. We're repeating that pattern over and over again. So then we went into cellular respiration and photosynthesis, and you need to see it on the big picture, right? So if I have CO2, what reaction is going to make CO2, which is inorganic, right? CO2 is an inorganic molecule. What reaction is going to make it organic?
Photosynthesis.
Photosynthesis. Okay, if I have an organic molecule, what reaction is going to make it an inorganic molecule? Cellular respiration. Those are those two reactions in cycle with one another. So I need to understand the process of each of those reactions and the role they play, like in cycling out your carbon or things like that. Okay? You want the big picture of each of these reactions understood. Let's break those down. In photosynthesis, there are two big reactions. What are the two big reactions in photosynthesis? Light dependent and light. Another name for the light independent? Another name? Dark reaction. I want to know, where does photosynthesis occur? It occurs in a chloroplast. Okay? And the light reaction occurs on the thylakoid membrane. And the reason is, what's embedded in those thylakoid membranes? Chlorophyll. Or other pigments, right? You know other pigments besides chlorophyll. Those pigments are able to take light energy and transfer that to electrons getting excited at the reaction center. Where is the source of those electrons? Water. Specifically, what part of water? The hydrogen ions, right? The hydrogen. So when you do photosynthesis and you split water, the oxygen immediately gets released, yay for us, but that's a waste product for photosynthesis. In fact, they're concerned about it because what can happen as the oxygen level builds up? Yeah, it can compete with RUBP in a light independent reaction and cause, what's the name of that reaction? Photorespiration. Hence, probably the evolution of aerobic respiration, because it removed oxygen out of the picture by getting reduced with hydrogen and electron to form water in cellular respiration. Okay, so anyway, you split the water, oxygen gets released, you have hydrogen. We'll now dissect the hydrogen into what two things? Electrons and a hydrogen ion. Hydrogen ion we'll go ahead and keep inside of our green pancake towers. And the electrons will get excited as they get passed and passed and passed, going down an electron transport chain. One place along there, PQ, is able to take that energy from that temporary holding of that electron that originated in hydrogen, which originated in water, okay, that got excited by the sun. It'll take that energy and take hydrogen ions and bring them to the inside of the pancake tower, right? Inside the thylakoid membrane. So now we got the hydrogens from the water itself and now additional hydrogen ions in there. So we can use that hydrogen ion gradient for chemiosmosis. We've seen that before. The importance of a membrane. How would I recognize a membrane that was ready for chemiosmosis? What would be evidence of a membrane that's ready for chemiosmosis? What, say it. I think you're saying it.
Protein synthase complex?
Oh, you mean ATP synthase complex?
Yeah.
Okay, so now before I can use that though, what other evidence would I expect? If a membrane is ready to make me ATP, what would I look for? Yes?
Like difference in pH.
Difference in pH. A difference in hydrogen ion concentration on one side of the membrane than the other. Difference in charge. Okay, I would expect to find if a membrane was going to do some chemiosmotic synthesis of ATP, whether it was in photosynthesis or cellular respiration, I would expect there to be a difference on either side. And I could test for a difference in pH, couldn't I? I could test for a difference in voltage and charge. I could test for a difference in concentration on either side of that membrane, because that membrane is critical for the synthesis of ATP. Okay, so there should be some sort of pumps in that membrane that allow, in photosynthesis, PQ allows hydrogen ions to go from one side to the other side. But if I was looking at a membrane in a mitochondria along the cristae of that inner membrane, I'd be looking for reductase, cytochrome oxidase. I would be looking for proteins that will put hydrogens on one side versus the other. And then I will look on both of those membranes, whether it's cellular respiration or photosynthesis, I'd be looking for something called ATP synthase complex, a passageway for allowing those hydrogen ions to go to the other side, synthesizing ATP. Right? Those are all the things you would look for on a charged membrane that had that power to do that. Okay, so back to the light reaction. We PQ, we make some ATP. Yay. Why do I want that ATP?
Cellular respiration.
Okay, I'm in photosynthesis right now, but I would if I, yeah.
Oh, you did for the
Calvin cycle. That's what I was looking for. Okay? But so there's two photosystem, right? Photosystem 2 and photosystem 1. Photosystem 1 is where those electrons dump off at the end. They rode one roller coaster, but we're going to go up and ride another one. We're going to get them excited by the sun again. But this time we're not going to let them, that roller coaster stop. It got jammed halfway down. And you're like, oh, nervous, hanging. Okay? And we're going to give those high energy electrons that don't go to the bottom of the roller coaster, we're going to give those high energy electrons because there's potential energy, because it wants to go back down. We're going to give those to NADP to reduce some NADP. We've done our job. We now have 2 of the high energy molecules we need to synthesize sugar out of CO2. And we'll do that where? In our chloroplast. Out in the stroma. That space around those green pancake towers. And there, I need something else from the environment. What do I need? Do I need oxygen? No. What do I need? I need carbon dioxide. High levels of carbon dioxide. And I want CO2 to bind with a molecule. Who do I want it to bind with? RUBP. RUBP is a 5-carbon molecule. It hooks up with a 1-carbon CO2, but that's an unstable bonding, right? Because she's a, and so that's going to what? Break down. And so instead of having 1 6-carbon molecule, I end up having 2 3-carbon molecules, right? And now, but they're low energy. I can't do anything with these. Okay, so I want, this is PGA, okay, I want to do something with these. So how can I power these molecules up? I use some ATP, which I got from the light reaction. Use some, and when I say use it, I'm taking the electrons from NADP and I'm giving them to these 3 carbon units, and now it's oxidized. Yes?
So would NADP oxidized be a product of the Krebs cycle?
Yes! Which I then could recycle back in the light reaction again. Okay? And NADP and inorganic phosphate would be another product of it. And PGAL or G3P would be another product of it. Okay. So then I use those high energy molecules. I make these high energy glucose, but I can't walk away with all of that product. I got to leave some in there so this can keep going. So I leave how many behind? 5, and I take away 1. Then I take those 5 high energy molecules, which have the potential to be so much, and I'm going to recycle them back into who again? RUBP. And if I do this cycle twice, I will walk away with 2 halves of glucose, which I may or may not synthesize glucose with. I may just keep it like that, because then I'm partway through the cellular respiration. Because remember, in glucose, what's the first thing we do? Break it in half. I don't have to break it in half. It's already broken, right? So that makes me think about glycolysis now. So I think about, but I also want to go on photosynthesis. And we'll do everything we'll loop the songs. We'll do that one time, okay? So how am I for time? Okay. If photosynthesis isn't working out, due to what process? If there's high oxygen, what is that? Photorespiration. That's bad, because it oxidizes the RUBP. Oxidizes, burns it. I don't want to do that. I want to continue having RUBP hook up with CO2. So ways to avoid that. It could either be in time, right? You're welcome. Time separation to avoid it, or space, physical separation. So we learned about C4 plants who isolate their Calvin cycle in the bundle sheath cells right around the veins. And they isolate them with mesophyll cells, remember that? And so those mesophyll cells, there's a compound in there called, it's like a cheerleader, it's PEP. PEP will say, I got you. PEP is faithful, not tempted by the fruit of another. It only hooks up with who? CO2. It will escort CO2. It will become, that CO2 we will have it bind, fix it to PEP. PEP will escort it into a Calvin cycle so that it releases it, it can be used. Or PEP at night in cam plants will fix CO2 at night when you can afford to have your stomata open and you won't lose any water. Yes?
I remember on the notes it said it wouldn't be on the AP exam. Do we still need to know it for tomorrow?
Yes. If it's on my notes, you need to know it because it can be support for adaptations.
Okay.
Yeah. Okay, so that was photosynthesis, and this brings us to cellular respiration. Who does cellular respiration?
All cells.
And the first part of it always occurs where? So you don't need mitochondria to do the first part at all. And how many steps is that reaction?
10.
And each one of those requires a different enzyme. And to get that party started, what do you have to do? You have to put in 2 ATP. But that's okay, because what's going to happen when I'm done? Get 4 out, so in my pocket I get to walk away with 2. And when I'm done, I have 2 pyruvic acids, right? And I have a couple of NAD. Those NAD aren't going to do anything for me unless I have a place to cash them out. Somebody gives you 2 checks, unless you have a bank, you can't do anything with them. Where's my bank? ETC. Okay? Now, in order to go into that bank, what's your passcode to get into that bank? You have to have some what? Oxygen, usually. Right? Usually oxygen is what's going to take you there. So if I don't have any oxygen, I can't do anything with those NAD. I might as well oxidize them so I can reduce them again, right? So, hey, how are you?
Yeah, Steph calling. I was just coming by to say hello.
Oh, I thought about you. Doing okay? Of course you are. Of course you are. Okay. All right. So if I don't have any oxygen, I need to just oxidize that NAD. There's 2 basic choices. What are your 2 basic choices? I can either produce alcohol and carbon dioxide. Who takes that choice? Plants, yeast. Or I can produce
Lactic acid.
yes. Either way, the whole reason I'm doing it, though, the whole reason I'm doing fermentation is to do what? Oxidize my NAD. And why do I want to oxidize my NAD?
So then you have a way to
I can use it in glycolysis again, right? Okay. Now, if I have oxygen, I'm going to transition into the what?
transition prep.
Transition prep. Yes. Where am I going? I'm transitioning into the mitochondria. Okay, and so I will take those pyruvic acids, rip a carbon off, and replace the carbon with what?
Acetyl-CoA.
With CoA. And then it becomes acetyl-CoA. All right? So that gives me the way to feed into this cycle, the Krebs cycle. The Krebs cycle further oxidizes that acetyl-CoA and generates what for us? What's the byproduct of doing that Krebs cycle? I get ATPs, but that's not the big product. What's the big product I get out of that? Reduced NADs. That's the big thing. Because every turn I get at least 3 NADs. And I get a 1 week little what? FAD. But I do get an ATP out of it, but that's not my goal. I do get it. Now I'm done. I've finished it there's still more energy in there, but I'm done. So now I go to the bank. My NADs and FADs get dropped off at the top of the electron transport chain, except for the weak ones. Same story as before. That whole chemiosmotic gradient, that membrane with the difference on either side, of which I could find evidence to show you that difference. And then I use that to make ATP. The key part to making this bank work, though, is at the bottom of the chain. Who's accepting those electrons?
Oxygen.
Oxygen forming
Water.
Perfect. Okay, are you ready? Cellular respiration, aerobic, then we're going to do photosynthesis, and then we're going to do anaerobic respiration in that way, okay? Got it? All right. So we're doing cellular respiration, and then aerobic, photosynthesis, and anaerobic. What's the whole thing called? Cellular respiration. What's the first step called? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. What do you start out with? Glucose. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What are these called? 2-pyruvic acid. And you reduce some NAD and make some ATP. 2. Oh, I have plenty of oxygen. What should I do? Aerobic respiration. What's the next step called? Transition prep. Here we go. And you reduce some NAD and 2-acetyl-CoA, put 1 away. What's the next step called? Another name for it. How many times am I going to do it? Where am I? Where in the mitochondria?
Matrix.
Here we go. 2 + 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an NAD, reduce an NAD, and 2 + 4 is 6. Do the bit, get 5. Do the bit, get 4. Make an ATP, reduce an FAD, reduce an NAD, and stop. Gather all your reduced NADs and FADs and take them to the top of the ETC. Reductase, reductase, cytochrome oxidase, and you make some ATP. A lot of it, a lot of it, a lot of it, or a lot of it, a lot of it. Who catch electrons at the bottom? Oxygen forming water. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweet, sweet. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. PQ, and you make some ATP. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Who catches it? NADP. Forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. Ready? 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, PJL. And you're left with 5C3. Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP, use some reduced NADP on your 6C3. Take 1 away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Stop. Pick up your glucose. How many carbons? 6. What are we going to do? Break it in half. How many steps? 10. How many enzymes? 10. What reaction is this? Glycolysis. Who does it? All cells. Where do they do it? In the cytoplasm. How many steps? 10. How many enzymes? 10. Good. What are these called? 2-pyruvic acid. And you reduce some NAD and make some ATP. 2. Oh no, we don't have any oxygen. What are we going to do? Fermentation. How many choices do we have? 2. You and I make lactic acid. Makes our muscles sore. Plants and yeast make alcohol. Either way, you oxidize your NAD. Good job. Clarifying questions? Clarifying questions. Okay, you're super smart. What's important to get tonight? Sleep. Sleep is important. You'd be surprised how important. Half your test, hey, talk to your friends later. Talk to me now. Half your test is going to be what? Essays. And half your test is going to be? Okay, so I haven't given you all the potential essays, but I've certainly given you hints about things you could prepare for, and I've given you some potential. So you know how to prepare for that. Don't try to memorize answers. Try to then you can apply them to new situations. Half the test is going to be multiple choice. How can you prepare for multiple choice tests? Yeah, take multiple choice. Okay, use your textbook. You've got a whole bunch in there and you've got answers right there in the back. If you came in and looked over your test last time, think about what was your weakest area. Was it multiple choice? Did you second guess yourself? Did you change answers? Was it your essay? Were you not clear? Did you get off track? Did you get nervous? Okay, think about, was it timing? Analyze what you did poor, what you weren't excited about, and be better about that tomorrow. You have all the potential energy. You have very low entropy. You have lots of potential energy. I believe in you. Yes? No. Super frustrated. But let me tell you, what you have there, you're good. You have all the, it'll just be one more layer of resource. Yeah. Okay, anything else? Okay, do you know that I love you?
Yes.
Okay. Gave you lots of hints today, so do well. Prepare.